Okay, welcome back after our tea break. Our next presenter is Dr. Neil Fourie. Dr. Fourie is an animal production veterinarian with a private practice in Middelburg. He's also a specialist toxicologist who has also been awarded with the SAVA Research Award during his career as a specialist. So, Dr. Neil, it is a great privilege for us to have you on this platform that you can share your specialized knowledge with us. Thank you. And I will hand over to you now, and then you can start sharing your presentation. Thank you.
Thank you so much. Good morning, everybody. It's a great pleasure to be with you. Um, and I'd like to present to you my toxicology adventure, uh, and especially applied to dairy today. Uh, to start off with, we should have a insight of what our animals are exposed to. At the top, you can see they're exposed to concentrates, obviously, uh, feed additives, water, medication, and roughage. And we'll run through these things very shortly. Okay, but some of our animals, although most of them are kept in very good environments and on a roof, and that we still have animals that are grazing. And I just want to run briefly through this, that the very most important plant poisonings in South Africa, we would like to identify 8 of them, which is gifblaar, gousiekte, cardioglycoside, synesiosis, geel dikkop, vermeersiekte, lantana, and cestrip. Some of them don't have any bearing on dairy animals. For example, vermeersiekte, I don't think anybody milks in the Northwest Cape, but the others do have a bearing. So let's just quickly run through them before we come to some other serious important things. The most important plant poisoning in South Africa is still poisoning. And sorry to bore you with some chemistry. You will see at the top is the cardenolites with a 5-membered ring. That is plants that are of no importance. But the bufadienolites at the bottom, if you see the green, it's got a 6-membered double unsaturated ring. Sorry, toxicology is coupled to chemistry, and these are the plants of serious importance. In fact, the bufadienolite name comes from the toad, where you can see the parotid gland just above the eye, and that produces a bufadienolite. And this is the most important plant, most probably in South Africa. I had a call this morning, about a farmer that lost a number of animals. Tulp, Moria pallida, the yellow tulp, is a winter problem. Okay, it happens now. Animals are on harvested maize lands where there's a number of tulp species, and even in the natural grazing. So this is yellow tulp, Moria pallida. And here you can see, this is in my area, that it looks like a cultivated land where you can see it's an old land, and all the green that you can see is tulp. And if you look at the mountains at the back, you can see it is brown and that there's no grazing. So if you put newly introduced animals onto this grazing, they will die. So the main thing about tulp is newly introduced animals that are not used to it, animals that usually grow up on tulp field are averse to the toxin. Okay, about the clinical signs, there are 4 systems affected. For those farmers that listen, tachycardia means accelerated heart rate and arrhythmia. You get a slight bloat. And the most important sign that we recognize nowadays is weakness of the hindquarters. It might sound like a simple clinical sign, but it's extremely important. You can see this animal is slagging behind in its hindquarters, and then they have respiratory distress. If I find them in the field, you can hear them grunting. They've got lung distress. This is the distribution of all the cardiac glycosides. The yellow one is the most important one, and that is TOLP poisoning. Down in the Western Cape, we get all these other interesting things like the plakkies and krimpsiekte, and then we get slankop on the green side, on the western side of South Africa, and they all cardiac glycosides. What's a treatment for a farmer phoned me this morning and said, what must I treat my animal with? Activated charcoal is the only thing that works. We've done many trials at Onderstepoort. It doesn't help to inject with anything, and the dose is 2 grams per kilogram per os. That means through the mouth. Okay, and the dose is very important, and it needs to be activated charcoal. You can't use your charka that you braai with on a Sunday and mill it up. This is activated charcoal, and there's a specific science about it. Another very important thing is to leave the animal right there where you treated it. It can stay down for a week or 2. You must give it food and water. A once-off treatment is fine, but you must leave the animal there exactly where it was. Don't try and change it to a crush pen. It will die because of stress on the heart. Okay, let's go to our next plant in the Eastern Cape. People from the Eastern Cape, synesiosis. You can see this land is just full of synesio, and you can see the animal as well has got liver damage. And there's Synesio latifolius, ragwort or stagger's bush, dinsiekte bossie. The adult plant usually doesn't cause problems. And there's the pathology for the students, severe liver damage to these animals. The next plant is Lantana camara, a plant that is invading our whole country. It started off in KwaZulu-Natal, then the old Eastern Transvaal, Mpumalanga. You find them in the bushveld. It's all over. And it's spread by birds eating the berries of these plants. And here you can see a dairy animal that was affected by lantana. The liver is severely affected, and we get photosensitivity. That means they get sunburn. So this farmer I went to treated 10 animals because he thought they've got anaplasmosis of gall sickness, galsiekte, because they were yellow. And then he called me out after the 10th animal was dead, and this is what I found. And it was lantana poisoning, especially affects the white parts of the body. And in unpigmented animals, you will find that only the nozzle is affected. But let's focus on the dairy animals, and this is what they look like. So lantana is a huge problem in most parts of our country, and it is spreading. It's a weed, it's a proclaimed weed, and you actually not have to, you must not have it on your property, you must take it out. You get severe liver damage and even kidney damage, and these animals rarely recover. You know, you can treat them with activated charcoal like we do with tulp, or give them liver supportive therapy, but they usually don't recover. Then another plant that I've added to our list. We usually had only 7 important plant poisonings. Now we have 8. Is Cestrum labigatum, inkberry, ink bessie. So this plant is causing massive problems. You can see the berries on the left-hand side, also spread by birds, and it's a severe liver toxin. And I spoke to a vet in the Eastern Cape, and he said it's not a shrub there like we like to describe it. It's a tree. And in the old days, we used to find this only on the banks of the Vaal River. Now it's all over, and it's also spreading. It's used as a garden shrub because you can see the nice yellow flowers on your right-hand side, but that's where the spreading starts. And that's the pathology. Okay, kikuyu poisoning, especially for the guys on the Eastern and Western Cape, severe sporadic problem. It will go good for years on kikuyu, and then suddenly animals start to die. And there are 2 syndromes. This is kikuyu in the Eastern Cape, and the one syndrome is associated with the armyworm. When pastures get infested with the armyworm, 10 days later, usually, we get poisoning with kikuyu. And this is what the armyworm moth looks like, Spodoptera exempta, commando worm. It flies down with a high winds from the western parts of Africa, and it usually settles down in the Eastern Cape. And then they lay their eggs, and then you get the armyworm in abundance. They're usually there in millions on the kikuyu grazing. We know that the worms themselves are not toxic, and kikuyu grass itself appears to be non-toxic, but this syndrome really puzzles us. And I spoke to Dr. Johan Olivier in Alexandria, and they say that kikuyu poisoning is exception rather than the rule. So he gets kikuyu poisoning without any armyworms. So what has been done? A fungus has been isolated, Fusarium toruliosorum, and it's an endophyte. But even that has still not been proven. So it's a mysterious disease. Like I said, it takes about 10 days for the grass to be toxic after armyworms, and the grass retains their toxicity for 4 to 6 weeks. The clinical signs of this disease is neuromuscular signs. We talk about sham drinking. That means they stand with their mouths in the water, but they can't drink. Inability to swallow. And then on a post-mortem examination, we find these frothy contents of the rumen, just full of kikuyu, fluid-filled rumen. And if you scrape off the epithelium of the rumen, you will see the reddish thing on the left-hand side where there's a rumenitis. And although their stomachs or rumens are filled with lots of water, they are dehydrated. And the bad thing about this disease is that a high percentage of animals die. It's got a very high mortality, and unfortunately, there is actually no treatment for this disease. Okay, to control it, we need to control the worms, which is often very, very difficult. And we can, the last point there, we can test for toxicity by using tracer cattle, and they should graze for 4 days before allowing the rest of the flock back onto the pasture. Okay, and then we've got a syndrome exactly the same to the one where there was army worm infestation. It also causes ruminitis, but there was no army worms. So you can see why I say that this is a very mysterious disease and a very difficult thing to explain. Okay, we now think it's a Fusarium species that causes that. And like you know, with fungi, it's you can't predict that. You need correct conditions, temperature and humidity for a mycotoxin or for a fungus to grow and to produce a mycotoxin. And the current thought is that it is Fusarium torulosum. Chinquirichi for the guys in the Western Cape. The Star of Bethlehem, beautiful cut flower, one of the most toxic plants in South Africa. It's even more toxic than gifblaar. If you work out in grams per kilogram, cause severe diarrhea and blindness 7 days after the diarrhea has started. Okay, and then also again for the guys in the Western Cape, annual ryegrass toxicity on annual ryegrass produces staggers and with a high morbidity. Okay, now we've finished with the plants and let's go through fungal poisonings. So this is often, to my knowledge, overestimated. You know, I always tell the students, you know, if you go to a doctor and he can't make a diagnosis on you, he tell you it's a virus. If a vet can't make a diagnosis, he will tell you it's a fungus or a mycotoxin. But fungal poisons are very specific poisonings. So you can't just judge a feed if it's moldy. You must make a specific diagnosis. And by that I mean you need to culture the fungus and you also need to isolate the mycotoxin. Don't just guess what has got into that feed. And there's a couple of mycotoxins. I'm not going to go through them all, but I will tell you what I think that is really important. The mycotoxins reduce feed intake. It reduces nutrient absorption and impairs metabolism, and several other. The most important one is 0.4, suppresses immune function. But I want to return back to my point, is that in ruminants, there is no importance of mycotoxins except for a few, because the rumen has got the ability to detoxify many mycotoxins. So please don't ever phone me and say you've diagnosed aflatoxin in an adult cow. It's never been recorded in South Africa in the history. Okay. So if you want to do fungal analysis, you must send it to the Plant Protection Research Unit. It's an ARC Research Council Institute at Roodeplaat Dam. And there's the number. They can do fungal analysis. But the mere presence of a fungus doesn't mean that the toxin was produced. So you also need to do toxin analysis. And that can be done at the Toxicology division of the OVI. And we often also send samples to international laboratories. Okay, let's talk about some serious fungus. Ergot alcoholate poisoning, summer syndrome causes hypothermia, and dry gangrene or fescue foot in the winter, because the fungus produces a toxin that causes vasoconstriction. For the farmers, that means narrowing of the blood vessels. Therefore, in the summer, they overheat, and the winter, they lose their extremities. The fungus can even be seen with your naked eye. Claviceps purpurea, here you can see it on annual ryegrass, the black seeds that was infected by Claviceps purpurea. And even on this grass, it can grow on any grass. This was a case in the Free State, which I investigated, was Tefai. And here's the normal yellow nutsedge geel eentjie. And look what these things look like. Totally infected with Claviceps cyperae. Very similar to the other Claviceps. And this is what happened to these animals. It was summer syndrome, hypothermia. They couldn't control their heat because all their blood vessels are constricted. They were breathing with an open mouth, and they showed abnormal behavior, like even walking into dams just to cause to cool down. In winter, you also get this closing of the blood vessels, vasoconstriction, and you can get a tail or the classic fescue foot, where little calves lose their hooves because there's no blood flow to this dry gangrene or fescue foot. As I want to emphasize that you can not always think if feed is moldy, that it is toxic. I investigated these 2 silos on a farm. It is worth 6 million rands worth of maize for human consumption, and it leaked. At the top, it was moldy from the top to the bottom. We had to cut it open at the bottom to get the maize out. And we did a small trial. I sent it to a Roodeplaat. I couldn't identify any toxic fungus. I sent it to Honor Support for toxin analysis, and they couldn't identify anything. And we decided to feed it to 2 or 3 animals. But eventually, we fed it to 4,000 animals. And the feedlot said it's the best production that they ever had. So fungi produces severely toxic substances, but sometimes also very good substances like proteins. I mean, we all know where penicillin comes from, and even things like selenomycin and penicillin are all produced by fungi. So they produce very good stuff. So make a correct diagnosis. Aflatoxicosis, I'm going to run through this very quickly. It's really a disease of monogastric animals. And that's why I told you earlier, never phone me to tell me you make me a diagnosis of aflatoxicosis. Aspergillus flavus, and yeah, monogastrics. It occurs in pigs and poultry. The rumen has got a wonderful mechanism of detoxifying aflatoxicosis. So I'm emphasizing now things that are of no importance, but we need to know that. Then moldy corn disease, Leukoencephalomalacia, it's a very prevalent fungus on our maize crops. And again, Fusarium verticuloides, the old name was Fusarium moniliforme. You can see the moldy corn there on your bottom right-hand side. But this is, again, a disease of horses. It doesn't affect ruminants. It's very important to know. The plodiosis, yeah, that's of importance on harvested maize fields in the winter, especially now. We've just combined harvested here in Middelburg, and then when the cattle go on the stalks, they can get diplodia. It's a disease that's characterized by moldy corn, and animals go usually in a wide bite stance with muscle tremors, and eventually they go down. And your biggest disease that you can confuse it with is botulism. But these animals usually recover with good treatment. They can still swallow, and that's the difference between botulism and diplodocus. And if you care for them, give them water and food until they stand up, about 90% of them recover. As we know with botulism, about 90% die. So it causes paralysis, they go down, but if you must turn it like the left-hand slide on the sternum, and you must never let the animal down lying like this on his side. But here we are most probably now with the most important mycotoxin of all mycotoxins, Aspergillus clavatus, marookvergiftiging, sorghumbean, residue poisoning, a deadly poison. It likes to grow on digested material. Like here you can see this is sorghumbean residue, and you can see the blue fungus on your right-hand side. So let me tell you, if you see a blue fungus, you must take serious attention of that. It's highly toxic. And here there was a craze in South Africa to plant these seeds in a store with humidity, and on your left-hand side you can see the blue fungus on the maize kernels, a deadly toxic poison. It's an unknown tremogenic mycotoxin. What we mean by that is, well, we don't know the toxin, but it's tremogenic. It causes muscle tremors, but with a high mortality. Most animals die. And I've investigated many of these outbreaks. Okay, here's a experimental animal that we have produced at Onderstepoort. They show hypersensitivity and severe tremors. And this is what we would call in veterinary science almost pathognomonic. This knuckling over of the joints at the back. There's only a few diseases that can cause that. And you get this knuckling over during this outbreak that I investigated. And the reason for that is that the muscles in the back, you can see it looks like fish. It is completely necrosed and severe pathology in these upper back muscles. Okay, let's talk about gossipol. There's lots about gossipol, and I'm, as a toxicologist, usually fight with the pathologist. It's very good food. I know in the Western Cape they feed up to 3 kilos of cottonseed or cottonseed oil cake, and again a disease of monogastric animals, deadly in pigs. I would agree that there are some varieties of cotton that comes from the middle of Africa that's got higher content of gossipol, and we need to analyze that. But for most, it's a monogastric problem. Okay, let's finish off with the feed additives. The iodophores urea, salt, and the beta agonists. Now, I don't think I've seen in all my years at Honest to Poort more poisonings with anything else with the ionophores. Fantastic products, but if you make a mix-up in the mixing process, you will lose animals. There are the 6 registered products in South Africa: monensin, siloamycin, meduramycin, narasin, and lisalicid. Okay, it's mostly used in poultry as an anticoccidial, it's a coccidiostat, but it's registered in dairy animals at 20 to 30 parts per million to prevent rumen acidosis, bloat, ketosis, and acute bovine pulmonary edema and emphysema. So it is registered. But I've seen outbreaks where hundreds of animals are affected. And as a forensic toxicologist, I will most probably be in court in the next couple of weeks where 1,100 animals died. And I need to be there as an expert witness to testify in this case. Okay, the one thing you will know about toxicology is that there's a great species difference. That's the one thing you must know. Horses are extremely susceptible to ionophores. You never feed cattle or sheep food to a horse. Vice versa, you can do it. But toxicity results from feeding errors too high and mixing errors. I've had so many cases where I investigate a feed mill and they made a 10 times error. Just a computer fault. It's a hard poison and also affects the muscles, and it is highly toxic. I know of one, and some of your farmers might know the farmer in the Free State that actually lost his herd due to too high levels of monensin. So it is something that you don't play around with. With urea poisoning, for the students, there are only one thing I want you to remember about this slide, and that's urea in the rumen is metabolized by the urease enzyme to ammonia and CO2, and this temperature occurs at an optimum of 49 or a high pH of 7.7 to 8. You need to know that how to treat animals with urea. Okay, large infusions of cold water to take the temperature of the rumen down, that reaction can't go on, and then obviously vinegar to bring the pH down. Water. We in South Africa, we are a water scarce country, and every farmer should analyze his water every year at a reputable laboratory like the CSIR for a complete analysis, because our water is changing all the time. This year we had a very good rain year, and it's better, but in dry years, everything concentrates like nitrates, cyanide, even fluoride. And you need to know what's going on your farm with your water supply. And you need to do that on an annual basis. Medication. We use lots of medication, and it is extremely important to see how these things are applied. I've been to a feedlot a couple of weeks ago, and they inject about 6 vaccinations plus minerals all on one site below the skin in the neck. So we need to look carefully how our medications are applied. This was a farmer photograph I took a couple of years ago, or as you can see in the photograph, many years ago, uh, when Bayer launched their first boron, and this guy thought this drug is too expensive. He will make his own boron, and he used an organophosphate in some oil and use it on his 20 dairy cows in the stable whilst they were milking. And as he was pouring on to the last animal, the first one was already dead. So you don't play around with chemicals. You use it according to the label and consult your veterinarian if you don't know how to use it. Okay, chronic copper poisoning I've included here because it's actually a sheep problem. And I'm going to run through this very quickly. But there are two dairy breeds that are susceptible to copper poisoning. Cattle are not susceptible to copper poisoning. They're very resistant, and I did that research. It's a sheep problem. But Guernseys and Jerseys comes from a specific island, the Guernsey and Jersey Islands, where there's a copper deficiency, a severe copper deficiency. And these animals are genetically bred to accumulate copper if they can get it. So one of the biggest outbreak of copper poisonings I've investigated in Jerseys, in Scotborough, one of the top breeders in South Africa, it was only Jerseys that died. So just be aware that Jerseys and Guernseys accumulate copper, but it is mainly a sheep problem. There you can see it, Jerseys and Guernseys. And the mechanism of the poison is that sheep accumulated, or these Jerseys and Guernseys, over a period of time. It's called chronic copper poisoning, but just suddenly one day, the liver starts to eliminate all this copper into the bloodstream, and you get all the red blood cells dying off. And then you get these yellow carcasses, absolutely yellow carcasses, because of all the red blood cells that have died. And then, just for interest's sake, we get what we call gunmetal black kidneys. And it's because of all the red blood cells that have died down, and red blood cells contain iron, and it's trapped into the kidneys, and then you get a gunmetal black pigmentation of the kidneys. Isn't it a beautiful description? And there's an enlarged liver, severely enlarged and yellow because of all the copper in there. Okay, I'm finishing off with the mother of all toxins, and that's botulism. Botulism is a toxicity, it's not a disease, because the organism that produces the toxin is Clostridium botulinum. And as a toxicologist, I want to just describe this very briefly to you. We can't even work out a lethal dose 50. It's too toxic. We talk about a medium toxic dose, which I don't want to go into detail, but that's a dose. So it's nanograms per kilogram, 0.388 nanograms per kilogram. Now, what is a nanogram? If you take a kilogram and divide it by 1,000, you go to grams. If you divide it by 1,000, you go to milligrams. If you divide it by 1,000, you go to micrograms. And if you divide that by 1,000, you go to nanograms. So for a 400 kilogram animal, I've made the calculations here. You need 0.155 microgram total days to kill an animal. Now, I've weighed these urea granules. They, on average, 4 milligrams. So if you divide that dose of botulism toxin into 4 milligrams, it is good enough to kill 25,000 animals. That's why there's a vaccine available, and it should be used on an annual basis. I've investigated deaths, mortalities of botulism of hundreds of animals. One time in Ladysmith, 200 animals died in 3 days. I fed the chicken manure at Onderstepoort to an animal. I gave it 1 teaspoon. It was dead in 3 hours. Acute death, without even showing any signs of paralysis or going down. So that is a compulsory vaccine. I will never blame a vet that misses the first cases of botulism, because I will show you my last slide. The toxin is in the dose. The animals that got in a lot, they die acutely. And only 3 or 4 days later, you see, okay, these animals are now lame and they can't get up. That's the animals that got the least toxin in. And these, the first ones, can die per acutely. You can just find them dead. And there's no pathology on a post-mortem in there. But only a few days later, you will see, okay, now it's botulism. So, okay, this is my last slide. I'm a toxicologist. The father of toxicology is Paracelsus. He lived in the 16th century. And he coined this phrase, sola dosis facit venenum. That's in Latin. And Latin is not that difficult. Sola means only. Dosis, the dose. Facit, facilitates or determines. Venenum, it's a poison. Only the dose determines the poison. So as a toxicologist and a forensic toxicologist, I'm often in court and as an expert witness, and we work out the dose. We don't guess. And like I told you with the mycotoxins, it is extremely important to not guess. If you can't make a diagnosis, don't guess. If you are involved with a severe case of poisoning, and I'm often involved with that, like this case we're going to do now with 1,100 animals that has died, which I think is malicious poisoning and not ionophore, because I've got all the results. Consult a toxicologist or a pathologist to assist you, because let me tell you, it's not always nice to stand in court and to be cross-examined. I thank you for your time.
Thank you, Dr. Neil.
Yes.
Are you finished?
Sorry, I just need to push up the volume here. I don't know how to. Okay. Okay, just say that again.
Okay, now I said thank you for your presentation. Are you finished with your presentation now?
I'm done.
Okay.
You know, my presentation was a 1-year course in toxicology that I tried to present in 1 hour.
Thank you so much. We got one remark here saying thank you, Doc. So if you have any questions, the participants, please put it in the chat box, or you can raise your hand to the icon and ask the question yourself. You've got another thank you saying it was very informative and interesting. We will just wait some time, Dr. Neil, for the people to ask questions.
Okay.
We still have another 15 minutes to ask questions before the next speaker will be here. Another person said, very interesting. Thank you for an awesome presentation.
Okay, just a remark from my side. I hope we can train more toxicologists in our country because as far as I know, we are only 2 registered specialist toxicologists, and it's an unbelievable field to work in.
So there's definitely scope for people to specialize in toxicology. You've got another thank you. Okay, Doctor, do you work with the research labs of Onderstepoort or do you work independently?
Yeah, I'm an independent toxicologist. It's actually a good question, but when I deal with a case, I'm a private toxicologist. If I deal with a case, you need to know where to send your samples to, and that has become a very big problem. I worked for 12 years at Onderstepoort Veterinary Institute, and we had a very good laboratory, but unfortunately, some of the tests and facilities just went south. At Onderstepoort, as we all know, we are sitting with a vaccine problem at the moment, but that's not the institute, that's OBP. It's a massive problem for us in private practice. But therefore, if you deal with a toxicity, you need to know where to send it to. And I've got several laboratories that I make use of because these cases end up in court. I can just tell you the case, and like I told you, I've been in court many, many times. And these dossiers end up on my table 3 years after the incident. And then I need to study it to make an expert opinion in court. And I can then see what the vet did, what the laboratory did, who did they send it through, what's the analysis. And that is my job, to study these dossiers. And like I said, it ends up on your table 3 years later because that's the lag period of our high courts in South Africa. And every case above 200,000 must go to high court. And the one I'm dealing with at the moment is millions upon millions. And then, it is sometimes embarrassing to see how the examination was done, what samples were taken. I went to a case in Grahamstown where a farmer lost 110 animals. The vet, which is a colleague of mine, took the brain of one animal as a sample. That was all samples he took. So now I need to testify against this vet, a colleague of mine, in high court. You know, it is embarrassing. So if you are in trouble, you need to phone somebody that knows how to handle these cases, because unfortunately in toxicology, all can go well for many months. But in toxicology, if something happens, it's usually of huge scale. Like this case I'm dealing with at the moment, where 1,100 animals died. Thank you.
Okay, thank you, Doctor. Then this person said he don't know whether you covered it because he joined a bit late, but he said he has an animal down with tulip.
Oh yeah. Okay, I did dealt with it, but I'll repeat it if you joined late. Okay, yeah, it's a good question. It's this time of the year. Like I said, he missed it now, but in my presentation, I spoke to a farmer this morning that I helped him with. So tulip pulp is a winter problem. It occurs now and they go down, and the treatment is 2 grams, not milligrams, 2 grams per kilogram of activated charcoal per os, and you leave the animal right there where it is down. Don't try and chase it up. It will die. Leave it there. They can stay down for a week, and you give it food and especially water if it stays down. The treatment is usually once off. You don't have to repeat it, but that's treatment.
Okay, thank you. Another question. Often toxicology is a race with time, and it seems the start point for proper diagnosis is what was done lately or fed to the animal.
Oh, sorry, can you just repeat that? I couldn't hear that properly.
Okay, it's actually a comment. Often toxicology is a race with time. It seems the start point for proper diagnosis is what was done lately or fed.
Okay, let me answer that. There are 5 pillars how to make a diagnosis, and the students should write this down. This is extremely important, which I didn't cover in my presentation. The one is a complete history. And what I mean by that is a complete history. You write everything down. You go and sit on that farm and you discuss it before you rush out. The next thing is pathology. You do a complete post-mortem examination and take samples of everything in formalin and on ice and freeze it. You can always throw it away afterwards. But if you don't have the samples, you don't have nothing. And that includes the feed samples. The third thing is obviously chemical analysis. And that's when I told you that if you're in trouble, you need to contact somebody where to send the samples through and take your samples. What usually happens in a big poisoning and where a feed company is involved, they will come and remove all the feed within 24 hours. And then there's no evidence. So you need to do that. Okay, and the last thing I want to mention, I think I skipped out on one thing now, is obviously laboratory analysis. The last thing I want to mention is experimental animal diagnosis. Now, in the old days, we had free access to animals. If we suspected a poisoning, like when I diagnosed that seleniumycin poisoning in the racehorses at Kailami, we just fed it to a horse at Onderstepoort. And it died within a day. But you can't do that anymore. we've got ethical committees and all sorts of things. But I do many clinical trials on a farm. But I have the background and the knowledge. Let me give you an example. If a farmer says, this fenbendazole, okay, which is Panacure and many other trade names, I know it is safe at 500 times the dose. Okay, and when he claims that this Panacure or fenbendazole, the active, kill these animals, I tell him, okay, all those 2 or 3 or 4 animals on your farm at 4 times the dose. And obviously it's proven then that it didn't die. So I make use of clinical trials, but on my own basis, because I'm not linked to a facility. If you're linked to Onderstepoort or one of these facilities nowadays, it will take you a year to get permission to do a trial. So therefore, but I won't do it indiscriminately. Not at all. If I know that there is a poison in there, I won't feed it to animal. But it's just often to prove the opposite, that there's nothing wrong with the remedy that you dosed.
Okay, thank you, Doctor. You've mentioned earlier that Cestrum is now spreading everywhere. And I want to know if you have noticed changes in the distribution of any other toxic plants, especially among the 8 important syndromes.
It's a very good question. Now, I think the, things like gifblaar synesiosis, vermeerziekte, that won't spread. It's very contained in a specific habitat. But the 2 things that do spread is lantana. I live here at Loskop Dam in Mpumalanga. It's just covered with lantana. That's spread by birds. So the things that are getting spread by birds are continuously spreading, and that's Cestrum and lantana. For the rest, I've mentioned, that will not spread at all. It is very much contained to a very specific habitat. But Cestrum and lantana is spreading, and we've got a massive problem with that.
Okay. And then, can you please assist with the name of the poison where a calf loses its limbs?
Okay, it is the mycotoxin Claviceps purpurea that causes that.
Okay, then another question. I don't know whether I will pronounce it correctly, but the Moraya pallida okay, it's a LD50, seems to also depend on dose ingested, but it seems to last longer in the body, as seen in feedlot cattle backgrounded in tulp-invested farm. How helpful is CBC in diagnosing pulp toxicity?
Sorry, I don't know that last term that you've mentioned, but yeah, like I said in my last slide, everything is dose dependent. But let me tell you, this time of the year, when tulp is green, it doesn't matter whether it's feedlot animals or whatever. If they take in, say, 1 gram per kilogram, I think that's more or less the toxic dose. They will most probably die. So the toxin is in the dose. And often the animals that recover did not take in, luckily, not enough toxin. But like I said, the only treatment is activated charcoal. And this time of the year is extremely important to keep, even on backgrounding. I've lost a lot of animals last year on backgrounding. So this is the time of the year to look out for. You must walk through those grazing. And we also find it on harvested maize lands, like it looks like cultivated. So this time of the year, I mean, you usually get tulp-free camps and tulp camps. You must go and walk through those camps and see if there's tulp in. Because especially with backgrounding, you're buying in animals from all places, and they're not used to tulp, and they will die.
Okay, I just want to say that the previous question, the CBC, referred to the complete blood count, whether that help in diagnosing.
Okay, yeah, no,
Yeah.
There's no blood count for tulp poisoning,
Okay.
nothing whatsoever.
Okay.
So we used to, when I was still at the faculty, it's actually a very good question. We could determine the cardiac glycosides. So you've got somebody clever who's asking this question. We could determine the cardiac glycoside levels, and we made use of a human laboratory like Dubuisson or Ampath or Lancet or whatever, because they test for digoxin in humans, which is also cardiac glycoside, obviously, to titrate the dose if somebody's on digoxin for cardiac failure or for whatever. And as we all know, it is such a highly toxic drug, like tulp is, that if you overdose, you will kill the person. So these medical laboratories have got a test method which is still available. And we used to send samples to them. We also used to extract liver and kidney for cardiac glycosides and send it to them to make a diagnosis, because it's often very difficult to make a diagnosis. I don't think the OVI, in fact, I spoke to them the other day, they don't do that test anymore, but it will be very interesting. But the only thing I can advise you, if you do send in samples to a medical laboratory, send in some control samples as well that you can compare it with. Because to be honest with you, I don't know what the normal values are. But please don't ask me these difficult questions.
Okay, then the last question we have is gifblaar a widespread problem or just in certain areas? And what treatment is available for gifblaar?
Okay, that's a very good question. Fairly isolated to the bushveld, north of the Magaliesberg. The closest gifblaar to Onnespoort is in Pretoria North. And then it occurs in the bushveld areas. Extremely, I didn't cover that because I was covering dairy animals. And, you don't get dairy animals in the bushveld. But an extremely interesting plant. It's an underground tree that only the leaves stick out on top, and we get poisonings in October, September, October, and again in March. And it's the Capitulum cymosum is the active principle, is a scientific name, and the principle is monofluoroacetate. No treatment. No treatment. We did all trials at Onnespoort with activated charcoal, with anything. Activated charcoal is a very interesting product. It binds certain molecules like cardiac glycosides very effectively, but some molecules not at all, like monofluoroacetate, 1080. It doesn't bind it at all. It binds gold. They recycle the old mine dumps for gold using activated charcoal. So gold is a small molecule and it binds that, but it doesn't bind monofluoroacetate. and we don't know why. But anyway, yeah, to answer your question, very fairly isolated areas where we get gifblaar. I'm here in the middle of between the high veld and the low veld, so I see everything. You know, on the high veld we get tulp, down here we get gifblaar. But let me tell you, if you chasing animals in a gifblaar camp, I know of one farmer last year who lost 40 cows dead of gifblaar. And I've got another farm here who lost 110 sheep. They just left the gate open to the gifblaar camp. So unlike tulp, where animals get feed aversion to tulp, they never get aversion to gifblaar. If it is September, October, and it's dry, and the tulp, the gifblaar is out there green, they will eat it, and they will die. 110 animals. You can take my email. I'll send you some photographs. No.
Thank you, Dr. Neil. It seems as though that is the only questions we have. Thank you so much for your participation and your briefing. It was extremely interesting, and I hope we will see one another soon in future.